Mudsnail Collaborative website. And you can also find access to the webinar by emailing info@nzmscollaborative.org. And with that, I'd like to introduce Sam Tank. Sam is the program specialist for the Great Lakes Commission Aquatic Invasive Species Program. Her role is to coordinate the Great Lakes Phragmites Collaborative and Phragmites Adaptive Management Framework. Sam works with land managers across the Great Lakes Basin to facilitate effective and efficient Phragmites management. Prior to joining the commission staff, Samantha attended Michigan State University, where she earned both her bachelor's and master's degree in fisheries and wildlife. And her master's work focused on conducting a social and biological evaluation of New Zealand mud snails in Michigan rivers. And her talk is titled Evaluation of a Qualitative Survey for Early Detection and Monitoring of New Zealand Mud Snails. And with that, I will mute and let Sam take over.
Thanks a lot, Jeremy. Just real quick, can you see my screen still as I advance to the next one?
Yes.
Okay, great. Please interrupt me if there are any issues with audio or visuals, but thanks everyone for joining us today. I'm happy to be the first presenter of the New Zealand Mud Snail Collaborative's webinar series. And today I'm going to talk about some work that I did in collaboration while I was at Michigan State University in collaboration with Michigan DNR and a couple other state agencies. We recently published a paper. It's entitled the same as my presentation, but I am going to expand on that a little bit. I would like to acknowledge my co-authors, Seth Herbst at the Michigan DNR and Dan Hayes at Michigan State University. And then beyond my co-authors, the funding for this project did come from the Great Lakes Restoration Initiative, and I do want to thank my partners at Eagle as well. So as this is the first webinar, I feel like it's appropriate to give a little bit of background on the New Zealand mud snail. So that's where I'll start out. So as you can see from the last image and this image up on the screen, they're quite small, about 2 to 6 millimeters in length when they're full grown. They are parthenogenic, so the populations that we're finding all over the United States really consists of very few clones that all originate from a single or just a few females. And so most populations are made up of females that reproduce parthenogenically. They can reproduce sexually as well. They are highly fecund. They reproduce multiple times a year, and their brood size can be over 100. They're small and quite cryptic. You can imagine, you can see they're dark in color. Finding them on river bottoms or amongst sediments can be tricky. And they are quite hardy as well. They have been known to survive days, if not longer, out of water, especially if they have a moist surface. And this really allows for them to be moved long distances. And they thrive in a variety of environments, so not just lotic, but lentic environments as well. They have a long invasion history where not only have they been found in North America, that's more recently, but they have established populations in Australia, Asia, and Europe. And I guess I didn't mention this, but as the name suggests, they are native to New Zealand. So this map just depicts their invasion history across the United States. They were first detected in 1987 in Snake River, Idaho. And then they quickly were detected across the United States. You can see in the Eastern Great Lakes region as well. By 2010, we were finding most of the populations out in the Western United States, but more and more popping up in the Great Lakes region. And today, and by present, this is probably 2 years outdated, but we're finding them even more widespread in this region and out west. So there's some concerns with mud snails being found so broadly across the United States. Out west, they've been documented in river systems to grow to abundances of 500,000 per square meter. And you can just imagine they're just stacked up all on top of each other at that point and can do some serious damage to the system, can compete with native macroinvertebrates that are important for our desirable fish species. And they can even survive digestion by trout. This kind of goes to their hardiness level as well. And overall, they can cause a complete shift in dynamics of food webs. So these are some of the reasons why we're concerned about finding them in Michigan. So Michigan was a little bit more recent of a detection. So they've been in the Great Lakes since 91, I believe, was the first time they were detected. But we didn't know of any existing populations until more recently in Michigan rivers. They were first detected by an eagle, which they were previously a DEQ employee, in 2015 in the Pere Marquette River. So that was the first time that we found them here in Michigan. And when we think about the invasion process, species don't become invasive overnight, right? So I've tried to illustrate that for you with this series of steps and transitions in this figure. So an non-native species is going to be introduced to a new area. Once they're present in a new area, they have to overcome some sort of barrier, and that initial barrier is growth and reproduction. And once they are able to reproduce, they have localized populations. And then the next barrier becomes getting to new areas, so spread. And then they're present in secondary locations. And then with continued spread, we have widespread establishment. So an invasive or a non-native species isn't necessarily invasive. We do need to see some sort of harm, either ecological, economic, or human health. And we don't really know, or at least we didn't have a great idea early on in 2015, 2016, when this work started, about where we were. So our assumption was we were pretty early in the invasion process, at least in Michigan rivers. So with this assumption in mind, we had to come up with some objectives of what we wanted to do. And I'm going to talk about a few of the things that we ended up doing between 2015 and 2018 with you today. So first, we wanted to determine where in Michigan rivers these mud snails were occurring. And to do that, we had to develop some sort of standardized method for sampling because there wasn't anything really consistent in the literature. And then second, we wanted to gain a better understanding of anglers as vectors. We knew that these small invaders could be easily moved around, but what was the likelihood that it was going to be anglers? So I'll start off by discussing the standardized sampling methods that we developed. So when we went into trying to decide what was going to make for an ideal sampling method and our ideal survey method, we wanted something that was quick, easy to implement, that we could go out and it was, gonna not take us all day to conduct one survey. It also is important that it was cost effective. Obviously, as resource managers, we don't necessarily have a lot of extra time or resources or personnel. So something that was low cost was going to be important as well. And then another thing was timely results. So there's some surveys out there, especially the more quantitative ones, that are highly effective, but there's a lag in result time. So something that yielded immediate results was going to be critical. We also wanted something quantifiable with a high detection probability. So there was a high likelihood that when we went out, we were going to actually detect them when they were indeed present. And then ideally a quantifiable estimate of abundance will come out of this survey as well. So we had some candidate methods in mind. In the literature, we had seen quite a bit of work done with the Hess or stovepipe sampler systems. They're pretty similar in what they do. There was Serber samplers. There was a qualitative survey method that we couldn't find anything really in the literature that we might have to develop. And then there was also the potential to use eDNA approaches as well. We evaluated all of the methods, or what we thought was going to be the likely outcomes of them. And we wanted something that checked as many boxes as possible. And what we ended up with was this qualitative survey. So if designed correctly, it should be able to do nearly all of the things we wanted, except give us a really quantifiable estimate of abundance, because it is by nature a qualitative survey. I did do some work on Hess and stovepipe samplers, and then 2 different eDNA methods, but we won't talk about that in this presentation today. So we knew we wanted to create a qualitative survey, but now we needed to design that qualitative survey. So once again, we wanted something that was going to check all of the boxes possible and that could be implemented and evaluated simultaneously. So what I mean by that is, as soon as we go out and we finish the survey, we know the results. This is just a diagram of the survey that we ended up designing. So it involved 2 to 4 searchers searching independently, and we would have one survey area. So this river system, this is a river. This represents one survey area. And within that survey, in this example, we had 3 different searchers. And the searchers would go out and independently search for 20 minutes. And you can see that we tried to avoid overlapping areas when possible. They would have a central access point and then move in different directions. Each searcher independently covered up to 50 meters of area. So it was 50 meters or however far they could get within that 20-minute time frame. All searchers focused on nearshore areas, and that's just because it was more likely that we were going to find mud snails in those areas. And this was a wading survey, so some of the rivers that we visited, it just wasn't possible to wade across or wade in the middle. And so those nearshore areas is where we're having the best luck finding snails. So it was targeted in that respect. Each searcher was collecting a representative sample. So I think we had the first 25 snails found. And we kept those snails, and then we kept any native snails, or just at least one of each native snail that we found. And we kept all of those in ethanol and stored them in the lab and used them to confirm identification if anything came into question. So that was the survey. It took probably about 30 minutes to complete, because each time we had a collection event for mud snails or other snails, we did time or pause our 20-minute timers. We just used stopwatch. And one thing that was really independent, or that was really important, was that they were truly independent. Searchers did not correspond with each other or let each other know, hey, we found, snails at this location, because we wanted to make sure that search effort remained as consistent as possible. So we analyzed the results from our 2017 survey data. We went out and surveyed 12 different rivers. And I'm sorry, my dogs are barking in the background if you can hear it. That totaled 227 survey events with 504 searcher observations. And so this is what we used for our analysis. And you can see we focused on streams that were on the west side of the state, and they were all cold water trout streams of high value. So the data was collected in a pretty simple manner. We would record individually per searcher the abundance of snails found. So either none, a low abundance, which was 1 to 10 snails, a medium abundance, which was 11 to 100 snails, or a high abundance, which is over 100 snails. So when I'm talking about our results and I talk about different qualitative levels of abundance, this range is really for a new invader. It's not what we were finding out west, where there's 500,000 per square meter. For a rare invader, over 100, we were considering high. So this is what our data ended up looking like. We had a unique site ID for each of our survey events, and then 2 different detection reports. So one from searcher 1 and one from searcher 2. We didn't include any surveys that had more than 2 searchers for our analyses. And you can see that sometimes there was conflict in what searcher 1 found versus what searcher 2 found. So this sort of data really lend itself to an occupancy analysis. And before I go into that analysis, I want to share some assumptions that we had to make in order to do that. So we assumed that mud snails do not move in or out of the survey area during the time of the survey event. So during that 20-minute to 30-minute time period, that they weren't drifting or they weren't migrating up or downstream out of that 50-meter range. We assumed that all mud snails were correctly identified. And we tried to ensure this by collecting that subsample of them for later identification if it got called into question. We assumed that the probability of detection was equal across all sites, or there's some covariate that can explain the variation. This one was likely violated, and I'll talk about why we were comfortable with this violation in a little bit, but mud snails are fairly patchy in their distribution. So we might come across a log and it has 300 mud snails on it, and then 5 meters down, there would be nothing just because there wasn't as desirable of habitat. So it's possible that within a search area, you could have easily had different probabilities of detection just because of the patchiness that we were observing. And then finally, we assumed that each searcher had a probability of encountering snails during a single survey, or the same probability. And again, this was violated for similar reasons. So getting into the analysis of our occupancy, here is the equation we used. I'm going to walk us through it, but this is what we ended up using. We ran all of our data in an R program. And if anyone's interested in that code, I believe it is published, but I'd be happy to share it as well. And I'll just briefly give you an overview of what we're looking at here. So T is the number of searchers at a site. Capital N is the total numbers of sites surveyed. And dot is the number of sites where at least one detection occurred. And psi is the probability of occupancy. And then P is the detection probability for a single searcher. And then N sub T is the number of detections on the teeth survey by the teeth searcher. So this is the equation that we use, but just to walk you through what that exactly means. So here's a oversimplified look at our data. So we have in this table, you can see that there's detections made by searcher 1 and detections made by searcher 2. So there's 2 options for detection. Searcher 1 can either detect at a site or not detect mud snails, and same for searcher 2. So there were 91 cases where we had both searcher 1 and searcher 2 detecting mud snails. And in that case, we call that a full detect. There were 34 cases in which only one searcher, so either searcher 2 or searcher 1, detected mud snails, but the other failed to detect. So we call those 34 cases partial detections. And then there were 102 cases where neither searcher detected New Zealand mud snails. So those were non-detects. And we assume that some of these are likely true non-detections where mud snails were not present, but then some of these were missed detections. So we ran the results of our occupancy analysis, and we determined that a single searcher going out under the protocols that I described had about an 81 chance of finding New Zealand mud snails when they were there. And if you increased to a second searcher, the detection probability increased to 96%, which was encouraging. That's what we wanted. We wanted a really high detection probability. So we did run the model in some non-standard ways to explore the implications of abundance. Because remember, we had this potentially violated assumption where the probability of detection was not equal across sites. So we wanted to see, okay, how is abundance going to be impacting our model results? So we ran the occupancy model first in two ways, where we included all non-detections and then excluded all non-detections. So those 102 times when both searchers failed to detect New Zealand mud snails. And then we also looked at the three qualitative levels of abundance, so low, medium, and high, and looked at how that impacted our detection. So we ended up running the model 7 different ways. So the first one was the one that included all of the data, and those were the first results that I shared. But the other 6, we ran the model in the way that's laid out by this chart. So including non-detections at each level of qualitative abundance detected, and then excluding those non-detections. And so we have a more conservative result and a less conservative result with running it in this manner. So this table provides a better breakdown of site classification data, and all of the survey sites that were classified as having a low qualitative abundance are shown in green. All of the sites that have a medium qualitative level of abundance in blue, and then high in purple. So you can see how that data really breaks down. So we ran all of these, and these were the results that we were seeing. So this overall up at the top, that included all 512 of our paired searcher events. And our occupancy, we have a relative estimate of occupancy here, and the detection probability for 1 searcher and 2 searchers. And so we have the results for all different levels of abundance. One thing I just want you to notice for this is the trend. So you can see that at low levels of abundance, when we include all of the non-detects, our detection probability with 1 and 2 searchers is fairly low, either 34% plus or minus with a single searcher, or about 57% with more searchers. But if we do that, and that's the more conservative estimate, if we run it without the additional non-detects included, so that would be kind of saying that all of those are true non-detects, and so therefore they don't provide any real value or information to the data, we would assume that occupancy is actually 100%. So all sites that were included were indeed infested. And the single searcher and the 2% searcher detection probabilities increase pretty dramatically. And you see this at all levels of abundance. And that goes up to when they're highly abundant at a site, you really have no chance of not detecting them, unless maybe your eyes are closed, I guess. So thinking back at what we were trying to get out of this survey, we were able to establish something that was rapid. It was cheap to implement. It yielded results immediately as soon as the those searchers, or however many searchers you used, were done. It was quantifiable, at least in the sense where we were able to look at those qualitative levels of abundance. It yielded a high detection probability, especially when you consider the data overall, but it didn't give us any quantitative estimates of abundance. So those were the standard methods that we implemented, but we wanted to look at the data and explore how could we potentially optimize the survey by changing different aspects of it. So searcher number is one thing that I've already briefly talked about, but what this figure gives you is it shows you when the data is all included, so all of those non-detects are included, or when the data has those non-detects excluded, and then the number of searchers. So this is really similar to what we just looked at. And again, this is just showing that trend that when mud snails are really rare in the system, our probability of detection is going to be quite a bit lower than if they're highly present or highly abundant in the system. However, overall, when we looked at our data sets, including all of the data, it really showed that overall, we still have a high likelihood of detection as long as we're implementing these surveys in the manner described. But there's also search time that we wanted to look at. So we were thinking about, okay, what was the, what's the ideal time? We just picked 20 minutes. It didn't necessarily mean anything. It was just a good starting point. What this figure is showing the time until first detection in minutes. So when did each searcher first detect that mud snail across relative abundance at the bottom? So there's low in blue, medium in purple, high in green, and then all of the data combined is showing up in this orange color. So we can see that even though it was harder to detect mud snails when they were in lower abundance, we were still finding nearly all of them within the first 10 minutes. And so this is the mean that's shown in those black triangles. So we did find them much quicker. It was, I think, about a minute or less when they were in really high abundance, and about 7 or 8 minutes when they were in low abundance. And overall, we were finding them generally in less than 5 minutes. But we did find some mud snails much later than that as well. So if you shorten the time, you are going to miss some snails. And so this figure just looks at it in a different way. This is the proportion of snails found, and this is across time until first detection. And again, it's broken up into high, medium, and low. So you can see that if we shorten the time to 5 minutes, you are going to lose all of these detections on the right side of the figure. And this just lays out the numbers a little bit in a different way in this table. So if you have a 20-minute time search under our protocols with 2 searchers, your detection probability is about 97%. If you decrease it to 5 minutes, you're really not losing that much of your detection probability if you keep with 2 searchers, only about 2%. So it's a consideration to make, because one thing that you can do if you decrease the time is you can potentially save resources and increase the number of sites. And there's kind of a lot to consider there when you're thinking about optimizing a survey, and we thought so as well. So what we did is we designed a decision support tool. So as others have observed, there's a high likelihood of missing an extremely rare species where true occupancy is 1% or less. And even when we're implementing the survey with high detectability at individual sites, if only a limited number of locations are surveyed, our overall detection probability is still going to be quite low. So as such, expectations for detecting new invaders really need to be set to realistic levels, depending on the number of sites that can be sampled. So this is the tool that we created to help managers make decisions. It can be modified for other surveys. It's not specific to New Zealand mud snails, but this is using the data and the detection probabilities that we were finding. So if we have, I'll just break it down for you. It shows survey events, so number of surveys that are conducted, and we have 10, 20, 50, 75, 100, and 200. And then true occupancy, this is something that can't be changed and just can be predicted. So this is really for rare species. So are they present in 1%, 5%, or 10% of overall sites that are looked at, and then site level detection probability. And these are the things where we can optimize to change our site level detection probability. So in general, overall detection probabilities at low relative abundances can be enhanced by either increasing the number of survey events or searcher replicates. So I just wanted to walk us through an example. So consider we're looking at going out and surveying 50 different sites. Well, we can assume maybe this is a really rare species and they haven't been found, very much in the system. So true occupancy is likely somewhere around 1%, extremely rare. We can look at increasing our site level detection probabilities by doing things like adding additional searchers, extending our search time, having more targeted searches where we're just looking in the areas where we expect to find that species. But you can see, even if we have 100% detection probability, which is not going to be likely, but if we have 100% detection probability, every time we go out, we're definitely going to find them. There's still a high likelihood that we're not going to detect them in the system. We only have about a 40% chance of detecting the species when they're present if we're visiting 50 sites. And that's due to them being extremely rare. So that's one way that we can try to increase our detection probability, but there's another way. We can also put our resources into sampling additional sites. So maybe we don't increase the number of searchers or the survey length, but we double the number of our sites. And so now we're seeing that, okay, doubling the number of sites about doubles our detection probability as well. So this tool is really just meant to help managers when they're planning their monitoring strategies and set realistic expectations of what they're likely to find or what those detection probabilities are likely to be given a certain amount of effort, and help you assess what's really needed in terms of effort and survey quantity. So that pretty much sums up that first objective. So I did want to move on quickly to the angler survey. And again, what we were looking at was, do anglers know about New Zealand mud snails? And if so, are they taking steps to decontaminate their waders in between fishing trips? And then are they traveling in between fishing trips? So even if they are showing risky behavior where they know about mud snails, but they're still not decontaminating, does it really matter? Are they really even moving between rivers? So this was following up an angler outreach effort that was a collaborative effort by the state, where signs were posted all over access points across Michigan to inform anglers about New Zealand mud snails in the spring of 2016. So we were able to conduct 308 angler surveys, and we just focused on the Pere Marquette. And these surveys began in early fall 2016, and we conducted them through late fall or winter 2017. And they were roving surveys in the sense that we weren't necessarily just stationed at access points. We would go out into the water, or we would walk angler trails. We would visit different access points. It was just kind of where we thought anglers were going to be in the system. We either surveyed anglers prior to their trip, during their trip, or once they were finished up. And we didn't discriminate between anglers who were waiting versus those who were in boats. We just recorded that. So one of the questions we asked was, were you aware that invasive New Zealand mud snails were detected in Michigan rivers in 2015? And again, there were these signs that were posted that anglers had to walk by. So we assumed going into this that there would be some level of awareness, and we were right. more anglers were aware than unaware. About 52% of anglers said that they knew about mud snails in Michigan rivers. They didn't have to have an in-depth knowledge. They just had to, have heard of them. But 48% were unaware. And so these were the signs. They were quite bright, stationed all over. Every angler that we surveyed definitely had to pass one. So these signs were they were doing something, but there was still room for improvement. They weren't catching everyone's eye, or at least not everyone was stopping to look at them. And then we asked anglers, did you disinfect your waders in between fishing trips? And what we found was that anglers were in general not decontaminating, and so more than they were decontaminating their waders. So what this figure shows is number of anglers surveyed, the proportion who were aware versus unaware, and then their behavior. So were they decontaminating in light blue or not decontaminating? And there wasn't a significant difference between those anglers that were aware of New Zealand mud snails and their decontamination habits versus those who were unaware in their decontamination habits. But the survey didn't really capture the reasons why they weren't or any barriers to action. It was really just looking to see what the relationship was between action and awareness. And we didn't find a significant one. But again, we had to ask ourselves when we were designing this survey, does it even matter if they're not a potential vector, if they're not traveling? And so we did ask anglers, have you fished another river within the last 2 days? If so, which river? Or do you plan to fish another river within the next 2 days? And again, which rivers are those? And we found that about 11% of anglers were traveling to other rivers, either in the previous 2 days or in the next 2 days they were planning on it. And this figure shows number of anglers, rivers, which I know is super small at the bottom, but you can see it's a variety of Michigan rivers and then also some out-of-state rivers as well. And the light gray bars, those show the next 2 days, and the black gray bars, the black bars show the previous 2 days. So anglers are pretty mobile. That means that those who are aware and not decontaminating, well, that's a behavior that we want to change because they are also mobile and moving to other rivers and could be that potential vector of spread that we want to avoid. So I'll just wrap up with some takeaways. Our qualitative survey that we designed provides good initial detection options. So this isn't something where you're going to go out and get a lot of information about the populations, but you will know presence, absence, and that's really where we need to start. It requires minimal training and is quick and easy and cheap to implement. So because of this, it really could be a good option to be implemented by participatory science groups. And I know that this survey is being used by the Michigan DNR in their surveys currently, but if local angler groups are interested in getting involved, these are easily trainable. The biggest, I'd say, barrier is being able to identify mud snails correctly. But once you can do that, and it's not necessarily that difficult, you can effectively implement this survey and then report back to a different managing body. In terms of our angler survey, anglers are likely vectors of continued spread, but there is this disconnect between awareness and behavior change. So our initial outreach efforts where we're informing people of the problem, asking them to take steps like clean, drain, dry, they're not necessarily changing behavior. And one thing I'll say about the angler survey is anglers who are decontaminating, we don't exactly know their reasoning behind why they're decontaminating. It might have nothing to do with mud snails at all, or it might be because of mud snails. So more work I know is continuing. There's some planned additional surveys that are looking at anglers and boaters planned at Michigan State University for the upcoming years. So stay tuned for those. So what we've learned over this time is that, well, we're likely a little bit further along in this invasion process than we initially thought. Our initial assumption, if you remember, we thought, okay, well, they were present in a new area. We didn't know if they had localized populations. We didn't know if there was secondary spread. But what we're seeing is that, yes, they have established populations and they are found in multiple locations. So what are we going to be able to do to prevent widespread establishment and prevent us getting to a point where mud snails are like they are in rivers out west, just everywhere at crazy high abundances? And our best tool that we have is going to be engagement and reaching out to anglers and other potential vectors and identifying those and figuring out messaging that really works to limit that spread and limit any further secondary introductions. But we also need to evaluate New Zealand mud snail impacts as well. And I know there's some work going on across the state that's looking at that, but my work didn't go into that realm of potential harm that you really need to have if a species is considered invasive. So I would say they are a non-native species from my observations, but are we getting to that level of harm? And I know others are documenting that. But in the end, really what we need is we need to develop long-term control methods if they are found to be harmful to our systems or changing food web dynamics. And that's something that we haven't gotten to at this point, but that is going to likely be an important next step in the future. So I'd just like to thank my funders at Great Lakes Restoration Initiative. I did receive some additional funding from the Forest Service and Red Cedar Fly Fishers. And then thanks again to my partner agencies. And finally to my field crew. We had a lot of fun going out and looking for mud snails. We found many thousands of mud snails in our time out there. And if anyone has told you that dogs can't be a part of your field crew, they are wrong. They were definitely valuable members of our field crew. So with that, I'll hand it back over for questions. Thank you.
All right, thanks, Sam. That was great. We do have a couple of questions. If anybody else has any more questions, please feel free to keep sending them into the chat. The first question for you is, what methods did you use to search for the mud snails? Like, how exactly were you looking for them?
Yeah, so we kept it pretty simple in terms of our equipment. We did use view ranger tubes or viewfinders. I'm going to forget the exact name now. They're basically long PVC pipes that have a plexiglass base to them, and you can look through them to see mud snails or to see, you know, the substrate at the bottom. We also did a lot of moving around the margins of the river, picking up vegetation such as any sort of substrate, but a lot of vegetation, woody debris, leaf packs, and just kind of combing through those quickly to see if we could find any mud snails that were adhered to those surfaces. Those are generally the substrates that we were detecting mud snails on, although gravel, sand, I mean, they really could be on anything. So we would make our way along the river margins for about those 50 meters and really just picking up substrate, looking through our view ranger tubes. Sometimes we use dip nets. We didn't find those to be as necessary. They took a little bit more time to sort through than we could just grab a handful of substrate a little bit easier. We did carry glass vials with us, and that's where we kept our mud snails that we were taking back for later identification. And after we got back to our central point, we'd kind of share the mud snails with the group, share our results, and preserve any found in ethanol.
Okay, Awesome, thank you. Second question, this one you, I think, touched on a bit at the end, but how dependent is the survey method on having expert or experienced searchers?
Yeah, I'd say not at all. So we had technicians who helped me implement the survey, and each year those were different. Sometimes we had, similar ones over multiple years, but it took probably a couple hours of training before they felt fairly confident. One good thing about this is you can collect additional snails and bring them back to whoever's in charge or coordinating the survey, and they can confirm those identifications later. So it's not something that if a searcher goes out and is inexperienced and makes a mistake, that you're not going to be able to catch that either. So pretty minimal training is involved in this, as long as you can identify mud snails and distinguish them from other snails in the river, and as long as there's no accessibility issues and someone can wade through the margins of the river. Pretty easy to do.
All right. A couple more questions. Is there any information on surveys done in streams and rivers of the southern portion of the state that are typically warmer?
Not to my knowledge. We did, I believe, most of between Michigan State, the DNR, and Eagle. I think all of the rivers that were surveyed were generally in the northern 2/3 of the state. There may have been some in the southern portion of the state. We did go into the UP, or Eagle did early on as well, but we didn't find any mud snails in those. And as you can see, we didn't focus on the east side of the state either. And we know that they're present, of course, in the Ausable River as well. So not that I'm aware of, but I'd be happy if you sent me an email to follow up on that.
Did your research or review include non-motorized boats such as kayaks and canoes as vectors of transport for the mud snails?
So we didn't include them into our surveys unless they were angling or they had been angling. So anyone in a, sit on top fishing kayak, we would ask them as well, but we didn't look at, we didn't ask them to inspect their equipment or anything like that. But I do for sure think that boaters are going to be a source of spread as well. And I think the survey that Michigan State University is planning on implementing in the upcoming years is going to take a look more at boats. And I'm guessing they're going to be motorized and non-motorized boats as well. One thing that I failed to mention, but I do want to just say quickly, is about decontamination practices. We did follow strict decontamination practices when we implemented the survey. We did a 20-minute Burcon soak of all of our equipment following each survey event. So even if we were staying within the same river, even if we were going half a mile downstream, we did continue to decontaminate all of our equipment, unless we were doing a floating survey using a raft where that wasn't possible. So that's something important to include, some sort of decontamination step if you are implementing this survey, because you don't then become a vector of spread yourself on top of anyone else who might be as well.
That's great, because that was actually one of the questions.
Oh, okay, good. I was just joking ahead.
Last question that I have, unless anybody else has some last minute ones to send in, is there any information as to what the highest densities are in Michigan and where do those occur?
I know that I believe Jeremy has done some more work on that. We haven't done, or at least I haven't done any work on looking at abundances, but I believe those are going to be coming out of different branches of the Ausable and then the Boardman rivers, because those are where, at least when I was still actively working on mud snails, where we were observing the highest densities, but I can't tell you exactly what those abundances are.
I got one more question pop up. Oh, Jeremy, do you have something to add?
Yeah, I won't take too much time. But yeah, you're right, Samantha. The Au Sable is where we're seeing the highest abundances thus far. And they're by no means what they're saying out west, but they've been steadily increasing early on in their invasions. And there's a lot of data and sample processing to occur still, but they're hovering around 50 to 100,000 per square meter in localized areas. As you mentioned, they're patchy. So depending on where you're looking. And just a quick note on the survey locations throughout the states as well and their distribution. There is we'll call it a draft map, but there's an interactive web map available on the collaborative website, which displays all the survey locations and the results of those locations in Michigan. And it'll show you where positive detections have occurred. And this is data that was compiled through DNR and MSU, Sam's work, and Eagle, as well as Charter Limited volunteers at Oakland University too. So it's kind of a clearinghouse of where surveys have happened, which should give you some insight on where they exist.
All right, couple more questions about decontamination. So I'm just going to try to kind of combine them. Why did you use Virkon and not Formula 409? And what methods of decontamination are most effective and least harmful for waders?
Least harmful for waders, that's a tricky one, but we'll start off with why we used Virkon. So we found that the Virkon solution was just one, it was the most convenient because we could fill up we used coolers generally, we could fill up a cooler, we could close the lid, and we could mix our 2% Virkon solution in that cooler, and it would be viable for a number of days. The solution does deactivate when it comes into contact with nutrients in the water, so it doesn't require any rinsing step in between and is safe, for the river system if there's residue left on our waders, because again, it would be deactivated by the nutrients within the river. And so it was really more of a convenience thing. We had, some pretty good assurance that mud snails weren't going to survive that 20-minute soak. We didn't need to mix up a new batch of anything each time, and we didn't have to have a drying step. And that's why we didn't use Formula 409. It is something that when we talked to anglers more anecdotally after the survey, when they asked us, generally decontamination did come up, about what they should be doing when they admitted that they weren't necessarily doing something, or maybe they were just rinsing with water or washing with soap or letting air dry, what were some other options. And a lot of them were fairly uncomfortable with 409 just because of the harm that it could cause to their waders, which, to be fair, they were also concerned about Virkon for that reason as well. And that it potentially needed to be rinsed from the waders before re-entering the river systems. And so in terms of what wasn't damaging to the waders, I'd say the most effective option is to have multiple pairs of waders and let them fully dry in between. Obviously, that's an expensive option because then you have to invest in multiple pairs of waders. Any chemical treatment is going to have some wear and tear to your waders. I don't think I saw any leakage for the 2 years that I wore my one pretty standard pair of waders and used the Virkon soak. It did discolor them slightly. So that's something to consider. But again, I wasn't rinsing or washing them in between. I was just using the Virkon and re-entering the river. So I'd say doing a complete dry and inspect is going to be the least harmful. Whether that's feasible or the most effective, likely not. 409 and Virkon are some of the more effective options out there.
Okay. Well, I don't have any more questions. You're getting some praise in the chat section, so Great job. And I'll turn it back over to Jeremy for some closing remarks.
Yeah, not too many. Thanks again, Sam. That was excellent, very informative. And to everybody out there, thank you very much for joining. There's more to come. Our next webinar will be in exactly 1 month on March 24th. This presentation will be recorded and made available through the website. And if you can't find it on there, just email info@nzmscollaborative.org. And if you have any follow-up questions that you're not thinking of right now, feel free to contact us at that same email or contact Sam directly. And again, thanks everyone. Have a great day, and we'll see you soon.
Thanks, Jeremy. Thanks, everyone.
Thanks, Sam. Bye-bye.